We'll move on to the next speaker. Effects of probiotic treatment on mucosal barrier function of GI tract of broiler chickens. Takahiro Nii, am I right? From Hiroshima University, Japan.
Okay, good afternoon, everyone. Today, I'm happy to have the good opportunity to explain my research in here. So today, I'd like to talk about the effect of the probiotics treatment on the mucosal immune barrier function in gastrointestinal tract of broiler chicks. So probiotics is known as have a good beneficial effect on the production and health of host animal, including the broiler chickens and layer also. So many researchers do study and reports the many kind of the data about the beneficial effect. For example, the increase of body weight gain and improve the FCR of them. And also, so probiotics treatment is known to enhance enhancement of the function of preventing infection against the pathogenic bacteria such as Salmonella and E. coli. So we are considering about these kind of beneficial effects are caused by the increase of immune function in the body. However, the detail of the probiotics effects on immune function is not yet clear. Okay. So these are the conceptual diagram of mucosal barrier. So this is a mucosal tissues. So mucosal epithelial are covered with mucus layers. This mucus layer including the secreted IgA and antimicrobial peptide. These kind of components are very important for eliminate the many kind of the pathogens. And also mucus layer is important as a physical barrier against the preventing the accessing of these bacteria from lumen to the epithelial cell. These mucus are produced by the goblet cells, exist on the epithelial cell layer. And these are the tight junction. Tight junction is one of the cell junction structure. So connect between each epithelial cells. This structure also very important for preventing the access, invading the pathogens inside to the tissues. So in this experiment, we are focused on the effect of the probiotics on the antimicrobial peptide production and mucus production and tight junction related molecules. Okay. So the goal of the study was to determine whether probiotics enhance the mucosal barrier function in the gastrointestinal tract of broiler chicks. We had 4 specific questions. One is structure of mucosal tissue of digestive tract changed by probiotics. And second one are functions of mucosal production, such as the frequencies of goblet cell and mucin-2. This is one of the mucus production related gene, so enhanced by probiotics. And number 3 is gene expression of tight junction related molecules, such as the claudin 1, 5, and zonula occludin 2, and junctional adhesion molecule 2, changed by probiotics. And number 4 is gene expression of antimicrobial peptide, such as avian beta defensin 2, 10, and 12, changed by probiotics. Okay. Oh, sorry. Materials and methods. We used the newly hatched chunky broiler chicks, and then they were separated to 2 groups. One is a control, another is probiotics treated group. And all of them were treated by oral administration, 300 microliter water with or without 5 milligram probiotics. And we used the commercial probiotics product. The name is FineLact, including the Lactobacillus reuteri. So these chicks were treated at day 0, 1, 2, 3, 4, 5, 6, and 1 day after of the last administration, they were killed and cropped duodenum, ileum, and cecum were collected. And these samples were processed to the paraffin blocks, and also RNAs are isolated from them. So paraffin sections were used for the two types of the staining. One is the hematoxylin and eosin staining. This technique used for the check of the morphology of the mucus structure, such as the thickness of mucosal layer in crop and villus height and crypt dips in the intestine. This intestine means duodenum, ileum, and cecum. Alcian blue staining also used for the stain for paraffin sections. So this technique were used for the check of the number of goblet cells in the intestine. And total RNA were used for the real-time PCR analysis. So we check the 4 types of the genes. One is the pro-inflammatory and anti-inflammatory cytokines. Second one is a mucus production-related gene. And third one is a tight junction-related gene. And last one is an antimicrobial peptide. These data are showing about the effect of probiotics on mucosal structure in the digestive tract. So in the case of the crop, we measured the length of mucosal layer like this. And in the duodenum, we are showing the data of the crypt dips, but not villus height. So why I didn't show the villus height is it is one of the problem in my experiment. I'm sorry. So that it means some villus are bended and fragmented. So in this case, difficult to calculate the length of the villus. So just now I show the crypt tips. And next, ileum, I measured the villus height and crypt tips like this. And in the cecum, in the case of the cecum, so this section are not clear, the junction of the villus and crypt. So in this case, I measured the total length of villus and crypt. So these data show the probiotics using in this experiment caused the improvement of the mucosal structure in ileum. And next. So these data shows pro-inflammatory cytokine and anti-inflammatory cytokine expression. So, but all of them didn't show any significant difference between the control and probiotics treated group in R6, 8, 10, and each segment. So it means this probiotics does not cause the mucosal inflammation in the digestive tract. So it means these probiotics did not have the bad effect on the intestinal morphology. Next. So this data shows the effect of the mucus production-related factors. So upper side means the number of goblet cells, and lower part gene expression of mucin-2. So this is data of the Alcian blue staining. And so this arrow means the goblet cell exist in the epithelial cell layer and in the crypt. We calculated the number of goblet cells in the epithelial cell layer and in the crypt. But there are no significant difference between control and probiotics treated group. And mucin-2 gene expression also, they didn't show any difference between the control and probiotics group. So it means probiotics using in this study may not enhance the mucus production in intestinal tissues. Next, these are the data of the gene expression of tight junction-related gene, Claudin-1, 5, and ZO2, and JAM2. So in the data of the JAM2 gene expression in crop and duodenum were significantly increased in probiotics treated group than control group, but not other parts. And I didn't show any mark in other data, but please focus on the Claudin-1 in the crop and ZO2 in crop. So they didn't have the significance, but they also have the tend to increase in the probiotics group than control group. So all of these data means maybe probiotics treatment upregulates the expression of tight junction molecules, especially JAM2. Okay. And this is data of the difference in gene expressions. So this is a difference in 2, 10, and 12. Difference in 10 in idiom and secum and difference in 12 in crop were significantly decreased in the probiotics group than control group. It means probiotics treatment don't regulate the expression of difference in 10 and 12 in digestive tract. Okay. These tables were a summary of results. This is data of the condition of mucosal tissues and mucus production and tight junction related genes and difference in gene expressions. So up mark arrow means higher in probiotics group, and down mark arrow means lower in probiotics group. In the condition of mucosal tissues, so only the structure of the idiom, such as villus height and crypt depth, were increased, but not have any information in these sections. And here, mucus production, there are no significantly difference or exist in this result. And here, in the tight junction, so JAM2 in crop and duodenum were significantly increased in the probiotics treated group. And last one, so difference in 10 in idiom and secum. Also, difference in 12 in crop were significantly decreased in the probiotics treated group. Okay. It is a conclusion of this research. Probiotics feeding using in this study may enhance the tight junction by upregulating the JAM2 gene expression in the crop and duodenum. But this probiotics may associate with the declining the ABBD difference in expression in the crop and intestine. Okay, so this part of the result in this experiment were analyzed by her. Her name is Jaisu Jirapat, one of the students in veterinary nursery in Kasat University in Thailand. So she was a very hard worker, and she is a very good, nice student. And she visited in our laboratory for 3 months, and she do work it. Okay. So I'd like to thanks to her, and I'd like to thanks to Thailand growing this kind of the very nice new researchers. Thank you. Okay.
Thank you. We may have one question if anybody asks. Yeah, please.
Sorry. Yeah. Thank you for your interesting presentation. May I know what strain of probiotics you were studying? Because there is numerous literature that also shows how probiotics can stimulate goblet cells to produce significantly more mucus, as well as the host cells to produce defensins. So is this referring to your strain? Because it's a sweeping statement to say probiotics does not. Maybe you should refer to your strain? Or?
So, I'm sorry, is your question is which cell produce the defensin and?
I'm saying that in literature, it's in published peer-reviewed papers in the journals, there are numerous articles that actually prove significant differences in goblet cell stimulation
Producing
and mucus production, as well as the host cells to produce antimicrobial substances such as defensins. So I think it's a bit generalized to say probiotics does not do this, but it's better maybe to refer to, was it the strain that you studied in this trial, or?
So in the normally, so this kind of the defensin exists so many type in the chickens. So the totally 14 type exist in the chicken. And some kind of the defensin, such as, for example, in this experiment, I use defensin 2. 2 is produced by the lymphocyte. But other type of the defensin 10 and 12 is produced from the component cell of the mucosal structure, such as exist in the some, but it is not clear. But maybe so this kind of some kind of the defenses are produced by the epithelial cell of the intestine, and also so other type defenses are produced from the component cell in lamina propria. And in this study, so the regress of the defensive expression. So we don't have the enough evidence of it, but I think so. We are thinking so maybe these degrees of the defensive expression are caused by the change of the microbiome in the intestine, because this probiotics is known as a change, improve the microorganisms in the intestine. So it means decrease of the E. coli and Salmonellas.
It's just an observation or comment from him, not a detailed query actually. His comment was, it will be a sweeping statement to say feeding probiotic does not enhance mucus production by goblet cells or defensins. It could be one observation in this particular study, but there are a lot of literatures to support it the other way. So it's just a comment of feedback, nothing else. Thank you very much.